Hi everyone, in this presentation we're going to be discussing the way nucleotides, meaning adenine, guanine, cytosine, and thymine, and ribose, how those are created, as well as how different pathways come together to make ATP used for DNA replication. So to begin, here on the screen is a poster that I've created over the past couple of weeks. It has a total of 15 different worksheets or parts to it. And I think it does a really good job of a whole as showing that a number of different pathways in the body are either interrelated or parallel to one another. So we'll come back to this poster as a whole again at the end and kind of highlight that once more. But right now I just want to draw your attention to a few of the different parts here to again show you how the nucleotides and ribose are made and then the ATP. So to begin, we'll take a look at nucleotides with worksheet 4. Nucleotides can be synthesized new for the first time or recycled through specific pathways, and the liver is going to be the major organ involved here for their synthesis. And so it's important to note that there's two different types here, the pyrimidines and purines, and they're going to follow two different pathways for their synthesis. So I'll blow up worksheet 4 here, and we'll be focusing right now at the top of this worksheet. The pyrimidines, again, that is thymine and cytosine, are synthesized first from aspartate, which is going to act as their amino acid nucleus, and then carbamoyl phosphate in the cytoplasm to the common precursor ring structure of orotic acid. And then onto this, a phosphorylated ribosyl unit is going to be covalently linked. And they're going to use PRPP as their source of their ribose 1-phosphate, which is shown over in that box, kind of in the lower left side there. And it's going to be important to note that unlike purine synthesis, the pyrimidines are synthesized as bases and then later are added to a ribose sugar, as in their ring is completed before they're linked to the ribose 5-phosphate. So that's what's kind of different between the two of them, which we'll touch on when we talk about purines in just a second. But shown up here at the top, this conversion first is going to yield us uridylate. And so we'll focus here on worksheet 5. That uridylate then that was created from kind of that first round there is going to act as a precursor for CTP and TTP. Again, we're up here at the top. The uridylate is going to be first converted to UTP, shown here in this box, via kinase reaction with 2 molecules of ATP. And CTP then is synthesized by the amination of UTP by the enzyme CTP synthase, also shown in that kind of big central box there. And this is going to give us cytosine triphosphate, or later then yield us the cytosine nucleotide. And then thymine in the cell will be synthesized as TTP from the UMP, going through methylation shown there. And so therefore now we have cytosine and thymine. We'll look at purines now, again back to worksheet 4. Now we're at the bottom here. Purines are first going to be synthesized from the sugar template onto which the ring synthesis occurs. So they're going to use glycine as their amino acid nucleus, and they are going to be synthesized as ribonucleotides. So that means that they have a nitrogen base, a ribose sugar, and a phosphate, rather than as just a free base, as the pyrimidines were. And both of the purines are going to be derived from the precursor IMP, so shown here at the bottom of this worksheet, as opposed to the pyrimidines that resulted in the uridylate. Now these purines so far, this first kind of synthesis part has resulted in isonate at the bottom, which is IMP. So we'll blow up worksheet 5. Now we're at the bottom of this one. That inosonate, or the IMP, is going to, it doesn't accumulate in the cells. Rather, as shown by a few of these reactions in this lower kind of central box, it's going to be rapidly converted into adenine, abbreviated there as ATP, and then the guanine, abbreviated there as the GTP. So at this point now we have all of our nucleotides, and so we're going to take a look now at ribose. So ribose in the body is a type of sugar normally made in the body from glucose. Ribose is going to play an important role in the synthesis of RNA, DNA, and also ATP. And the glucose used as the starting point can come from a few different places. So if we take a look at worksheet 8, shown here is gluconeogenesis, or the creation of glucose from the breakdown of dietary fats or fats stored in the body. Glucose can be created from here and used for the creation of ribose. Or if we look at worksheet 7, it can be used from just the dietary intake shown here at the beginning of the start of glycolysis. And so at this point, I want to draw your attention over here to worksheet 1, which is the pentose phosphate pathway. And so I'll blow that up here. The pentose phosphate pathway is going to generate NADPH and pentoses, which are 5-carbon sugars, as well as the ribose 5-phosphate. So this is where we're getting, again, our ribose 5-phosphate, which is later then used for building DNA and RNA. And I also want to note that it can produce PRPP, shown up there at the top. And that is what we use all the way at the beginning, talking about the synthesis of nucleotides, of how the pyrimidines needed that. So that's how this pathway then plays a role back into that. So quickly back to glycolysis of worksheet 7. With glycolysis, glucose metabolism occurs in the presence of carbon dioxide in all of our cells, and it's going to produce us, as you work your way down here on the screen to the bottom there, 2 molecules of pyruvate. And so this again occurs outside of the mitochondria, usually in the cytoplasm. And then just a quick side note here shown in worksheet 13 that I'll blow up. Pyruvate can be used to generate aspartate and glutamate, and those we use in the synthesis of the pyrimidines and purines respectively back when we talked about, again, the creation of nucleotides. That's again how this pathway is then interrelated to both of the other ones that we've talked about. But what we're going to kind of focus on here is the use of pyruvate in the citric acid cycle. So pyruvate up here at the top of this worksheet that I blew up is going to be converted to acetyl-CoA, and that's going to feed directly into the citric acid cycle at the bottom of the screen there. And then in worksheet 11 I'll blow up, from the citric acid cycle, the electron transport chain is shown here, and it displays the way the NADH and the FADH derived from the citric acid cycle are oxidized in the protein chambers, and then ATP is going to be produced in the mitochondria in the last protein chamber, known as ATP synthase. And so finally, our last worksheet for today is, I'm going to blow up worksheet 6 here, which is a kind of larger blown up depiction of ATP being created from the ATP synthase, or in that ATP synthase chamber. And the ATP is being kind of created here from the ADP and the phosphate. And this whole process gives us the bulk of our ATP in the body. So throughout some of these worksheets here, a little bit of ATP was either used or generated in a number of the other pathways, but the citric acid cycle to the electron transport chain and the ATP synthase chamber is going to give us the bulk of our ATP in the body, which was during aerobic cellular respiration and made by oxidative phosphorylation. So to conclude, this is just an overall shot of our poster again, now that we know how the nucleotides are created, how that ribose is made, and how ATP is made in the body, the bulk of that, for all these coming together then to later help with our DNA and RNA creation and replication. And so this is just a few select worksheets and kind of pathways that I pulled out. There's more here than just what I described. And again, by having a lot of these processes related, the body can work very efficiently and effectively to create that ATP, ribose, and the nucleotides described earlier. Because while we're always using ATP, we're going to get a much larger net gain of ATP. And it's just very efficient, again, for the body to have all these processes related. So I hope you learned a little bit more. and have a little bit of a better understanding of some of the processes and pathways in the body. Thanks.